Hi, and welcome to Reviewing with Mrs. Wages. Today we're going to talk about cell structure and function. So let's begin with objective number 1, which asks you to identify major cell structures and describe their functions. One of the most important parts of the cell is the nucleus. The nucleus is a membrane-enclosed structure in eukaryotic cells. It contains and protects DNA. And remember, the DNA is in the form of chromatin, which is loosely coiled chromosomes. The job of the nucleus is to control cellular activity, and it does this by regulating gene expression. This diagram shows the cell membrane. The cell membrane is the boundary of the cell, and it's made of phospholipids and proteins. It separates the cell from its surroundings. It controls what goes in and out of the cell. Here you can see the smooth endoplasmic reticulum. This organelle is described as smooth because there's no ribosomes on its surface. One of its jobs is to make lipids that are found in the cell membrane. This is the nuclear envelope. It's the boundary to the nucleus. This membrane keeps the chromatin separate from the cytoplasm. And as you can see, the nuclear envelope is also very porous, which allows some things to escape from the nucleus. This diagram shows you a plant cell, and it's focusing on the chloroplast, which is the site of photosynthesis. This organelle contains a pigment called chlorophyll, which absorbs light to drive the production of glucose, and animal cells do not have chloroplasts. In this diagram, the arrow is pointing to the centrioles. Centrioles are small organelles composed of microtubules that are found in most eukaryotic cells, except for plants. Centrioles help form spindle fibers, which help separate chromosomes during cell division, such as mitosis or meiosis. These arrows are pointing to ribosomes. As you can see, ribosomes are found on the endoplasmic reticulum as well as free-floating in the cytoplasm. They're composed of RNA, and they carry out protein synthesis, which means they make proteins. They're found in both prokaryotic and eukaryotic cells. This diagram is showing the small vacuoles found in animal cells. These are fluid-filled organelles in some eukaryotic cells. They store food, water, or other substances. This is showing you the cytoplasm found in animal cells. This is the liquid filling of the cells, and it contains the organelles and the nucleus. This is the rough endoplasmic reticulum. You can tell it is because it has ribosomes all over its surface. This structure produces the proteins that are to be exported out of the cell. It works very closely with the green Golgi body. This arrow is pointing to a lysosome. Lysosomes are membrane-enclosed organelles in animal cells. They contain digestive enzymes to break down food, particles, or eliminate waste from the cell. Here you can see a plant cell with a very large central vacuole. Again, these vacuoles are for storing water, food, or other substances. The focus of this diagram is the cytoskeleton, which is made up of both microtubules and microfilaments. It controls cell shape, cell movement, internal organization, and the transport of organelles within the cell. Think of it as the framework for the cell. This arrow is pointing to a single mitochondrion, which is an organelle found in eukaryotic cells that produces ATP in a process called cellular respiration. The plural for mitochondrion is mitochondria. The diagram on the left shows you the hair-like structures called cilia. The diagram on the right shows you the whip-like structures called flagella. Even though they're very different in structure, they're both important for movement of the cell. This green structure is the Golgi body. It modifies, sorts, and packages proteins that were produced by the rough ER. It puts these proteins into small vesicles to be transported out of the cell. You can think of the Golgi body as putting the finishing touches on these important proteins. This little kernel inside the nucleus is called the nucleolus. It uses RNA to begin the assembly of ribosomes. Make sure you know how to spell it correctly, and make sure you know that it's different from the nucleus. These small green structures are vesicles. They are membrane-bound sacs in the cell, and their job is to store and transport cellular products. Sometimes they merge with the cell membrane to export important things like proteins. This plant cell has a very rigid cell wall surrounding its cell membrane. It provides support and protection for the cell. Though it does not control what goes in and out of the cell itself, That's the job of the cell membrane. Plant and animal cells are both eukaryotic cells. They share very similar characteristics, though each one has specific unique structures. An animal cell, for example, has lysosomes, centrioles, and small vacuoles. None of these are found in plant cells. Plant cells, however, have chloroplasts, cell walls, and large vacuoles. And now we're on to objective number 2, which asks you to explain the cell theory. The cell theory is composed of 3 important parts. Number 1, all living things are composed of cells. Number 2, cells are the structural and functional unit of life. And number 3, new cells are produced from existing cells. Now it's easy to write these things down, but in the test you'll have to explain them in your own language. Provide good detail, give me examples, convey to me that you understand what each of these things mean. Objective number 3 asks you to compare prokaryotic and eukaryotic cells. Though there are many diverse types of cells, all cells fall into one of 2 categories. They are either eukaryotic or they are prokaryotic. This picture shows you both. The left is a simple bacterial cell, which is prokaryotic. The right shows you a complex animal cell, which is eukaryotic. To convince me that you understand the difference between these types of cells, you'll have to explain the structural differences between each of them. Let's begin with prokaryotic cells. These cells lack a true nucleus. They do have DNA, it's just kind of concentrated within the cytoplasm in the middle. It's not in a container like the nucleus. These cells are simple in structure. They have no membrane-bound organelles. They're typically smaller than eukaryotic cells, but not always. An example of prokaryotic cells would be bacteria. Eukaryotic cells are much more complex. They have a true nucleus. Their DNA is contained within that structure called the nuclear envelope. They are complex in structure. They have membrane-bound organelles such as chloroplasts or mitochondria. They're typically larger than prokaryotic cells, but again, not always. An example would be animal cells. The flashback portion of this test is going to review enzymes, so let's get started. What are enzymes? Enzymes are proteins which catalyze chemical reactions within cells. This means they speed up chemical reactions happening inside of the cells, and they do this by weakening the bonds of the substrate. These reactions happen quickly, and they happen at a lower temperature because of the enzymes. We don't need a lot of heat to make the reactions go quickly because enzymes are there to do the job. An example of enzyme that you're familiar with would be catalase. This enzyme is found in liver cells. The substrate from our lab was hydrogen peroxide. Your cells generate hydrogen peroxide, and with the help of catalase, they break it down very quickly because hydrogen peroxide is toxic to cells. That's all for now, and as always, happy studying!